Flowering plants undergo a unique reproductive process where there are two fertilization events. This double fertilization event occurs between the male reproductive organ, the male gametophyte, and the female reproductive organ, the female gametophyte. Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains one reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 of these megaspores degenerate, leaving only one surviving megaspore. This surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. This complete structure is known as the embryo sac. Within the established embryo sac, cell walls begin to form between most of the nuclei. 3 cells named antipodal cells form opposite the opening of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gamete, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains 2 main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the 2 sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.